Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on translation or protein synthesis. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. During the initiation stage of translation, mRNA first binds to option 1 small subunit of ribosome, option 2 large subunit of ribosome, option 3 tRNA, option 4 70S ribosome. And the correct answer is option 1 small subunit of ribosome. The first step of the initiation stage of translation is the binding of mRNA to the small ribosomal subunit through base pairing with appropriate sequences on rRNA. In case of eukaryotes, this small subunit of ribosome is 40S ribosomal subunit, whereas in case of prokaryotes, it is 30S ribosomal subunit. Okay. Next question. Which of the following is an initiation codon? Option 1 AAA, option 2 AUG, option 3 UAG, option 4 UGA. This A stands for adenine, the nucleotide base adenine, G stands for guanine nucleotide base, U for uracil. Okay. And the correct answer is option 2, that is AUG. AUG is the initiation codon. Translation starts with a chain termination codon or start codon and terminates with a stop codon. The start codon is the first codon of the messenger RNA, that is mRNA transcript, that is translated by a ribosome. Next question. The movement of a ribosome along mRNA in 5' to 3' direction is known as option 1 translation, option 2 proofreading, option 3 ubiquitination, and the option 4 is translocation. And the correct answer is option 4 translocation. During protein synthesis, mRNA and tRNAs are moved through the ribosome by a dynamic process of translocation. Sequential movement of tRNAs from the A site or amino acid site to the P site, that is peptidyl site, to the E site, that is exit site, is coupled with the movement of their associated codons in the mRNA. Next question. How many release factors are identified in eukaryotes and prokaryotes? Option 1, 1 and 3 respectively. Option 2, 3 and 3 respectively. Option 3, 3 and 2 respectively. Option 4, 2 and 3 respectively. And the correct answer is option 3, 3 and 2 respectively. That is, for prokaryotes, there are 3 release factors, whereas for eukaryotes, there are 2 release factors. In prokaryotes, the translation termination is mediated by 3 release factors, which are RF1, RF2 and RF3. Whereas in eukaryotes, translation termination requires 2 eukaryotic release factors, that is ERF1 and ERF3. ERF1 is required for the recognition of the stop codon, whereas ERF3 supports that polypeptide change release in a GTP dependent manner. So, in prokaryotes, there are 3 release factors, whereas in eukaryotes, there are 2 release factors. Next question. Ribosome is option 1 RNA, option 2 ribozyme, option 3 antibody having enzymatic activity, and option 4 is RNA having enzymatic activity. And the correct answer is option 4, that is RNA having enzymatic activity. And the ribosome is a ribonucleic acid enzyme that catalyzes a chemical reaction. The ribosome catalyzes specific reaction in a similar way to that of protein enzymes. It is also called catalytic RNA because it has enzymatic activity. Ribosomes are found in the ribosome where they join amino acids together to form protein chains. So, this ribosomes are RNA, that is ribonucleic acid, that with enzymatic activity. Whereas antibody having an enzymatic activity is known as AbGyme. Next question. In prokaryotes, first amino acid to be incorporated during translation. Option 1 methionine, option 2 N-formyl methionine, option 3 lysine, option 4 is glycine. And the correct answer is option 2 N-formyl methionine. Whereas in eukaryotes, the first amino acid to be incorporated during translation is methionine. The peptidyl transferase is located in option 1 small ribosomal subunit, option 2 large ribosomal subunit, option 3 tRNA, and option 4 is mRNA. Option 2 is large ribosomal subunit. So peptidyl transferase is located in large ribosomal subunit. The peptidyl transferase is an enzyme that catalyzes the addition of an amino acid residue in order to grow polypeptide chain in protein synthesis. It is located in the large ribosomal subunit where it catalyzes peptide bond formation. So it catalyzes the peptide bond formation. It is composed entirely of RNA. So this peptidyl transferase is located in large ribosomal subunit. So this large ribosomal subunit in case of eukaryotes is 60S ribosomal subunit, whereas in case of prokaryotes it is 50S ribosomal subunit. Okay. Next question. Which of the following RNA molecule act as peptidyl transferase in prokaryotes? Option 1 16S rRNA, option 2 23S rRNA, option 3 28S rRNA, option 4 5S rRNA. And the correct answer is option 2 23S rRNA. In prokaryotes, the 50S ribosome subunit contains peptidyl transferase component and act as ribozyme. This 50S subunit's 23S component has the peptidyl transferase activity. The peptidyl transferase centers on the 50S subunit lies at the lower tips, that is the acceptor ends of the A and P site of the tRNAs. Whereas in eukaryotes, the 60S, that is ribosome subunit, contains the peptidyl transferase component and act as ribozyme. Next question. The elongation factor in prokaryotes are option 1 EFTS and EFT2, option 2 EFTU and EFG, option 3 EFTU and EFT1, option 4 EFT1 and EFT2. And the correct answer is option 2 EFTU and EFG. The elongation factors are a set of proteins that function at the ribosome during protein synthesis to facilitate translation elongation from the formation of the first to the last polypeptide bond of a growing polypeptide. The common elongation factors in prokaryotes are EFTU, EFTS and EFG. So here the second option will be the correct, that is EFTU, EFG. The eukaryotic translation initiation elongation is mediated by two elongation factors, that are small EF1, small EF2. Next question. A group of ribosomes attached to the same mRNA-like beds on a thread is known as option 1 polypeptide, option 2 polysome, option 3 mesosomes, option 4 is lysosomes. And the correct answer is option 2 polysomes. A ribosome that is polyribosome or polysome or argosomes, is a group of ribosomes which is bound to an mRNA molecule like bits on a thread. It consists of a complex of an mRNA molecule and two or more ribosomes that act to translate mRNA instruction into polypeptides. Polysomes are formed during elongation phase. When ribosomes and the elongation factors are synthesized, the encoded polypeptide. Multiple ribosomes move along the coding region of mRNA, creating a polysome. Next question. Which of the following part of mRNA directs AUG to the appropriate position on 30S smaller RNA? Option 1 poly-A tail, option 2 shiny dalgona sequence, option 3 5' guanosine cap, option 4 none of the above. And the correct answer is option 2 shiny dalgona sequence. The shiny dalgona sequence is the ribosomal binding site in bacterial and archaeal messenger mRNA, that is mRNA. It is generally located around 8 bases upstream of the start codon of AUG. The RNA sequence helps recruit the ribosome to the messenger RNA to initiate protein synthesis by aligning the ribosome with the start codon. Next question. tRNA looks like option 1 clover leaf, option 2 L-shaped, option 3 inverted L-shaped, option 4 both 1 and 3. And the correct answer is option 4 both 1 and 3. Although the structure of tRNA appears clover-like, but in actual structure, the tRNA is a compact molecule which basically looks more like an inverted L. The molecule of tRNA is folded due to the folding of some complementary nitrogenous bases that come across with each other and form hydrogen bonds among them. So the answer will be tRNA looks like both in clover leaf and also in inverted L-shaped. Next question. The 3' end of all tRNA molecule contains which of the following nucleotide sequences? Option 1 3' ASC, option 2 3' CCA, option 3 3' ACC, option 4 3' AAA. And the correct answer is option 2 3' CCA. C stands for cytosine base. The acceptor arm is a 7 to 9 base pairs stem made by a base pairing of the 5' terminal nucleotide with a 3' terminal nucleotide, which contains the CCA tail used to attach the amino acid. The amino acid loaded onto the tRNA by amino acid tRNA synthetase to form amino acid tRNA and is covalently bonded to the 3' hydroxyl group on the CCA tail. Next question. tRNA molecules are identified on the basis of option 1 acceptor arm, option 2 anticodon arm, option 3 D arm, option 4 all of the above. And the correct answer is option 2 anticodon arm. The anticodon loop is situated opposite to the amino acid acceptive arm and is responsible for recognizing and attaching to the specific codon of mRNA. Next question. Transcription and translation can be coupled in option 1 bacteria, option 2 archaea, option 3 plants, option 4 animals. And the correct answer is option 1 bacteria. Coupled transcription and translation is a hallmark in prokaryotic gene expression. So this coupled transcription and translation is hallmark of prokaryotic gene expression. Coupled transcription and translation occurs when the ribosome associated with and initiate translation of mRNA whose transcription has yet not concluded, therefore forming RNAP. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.